Hello everyone, it is a pleasure to be with you today. Currently, there are approximately 39 million people worldwide living with HIV. Because of this, recent news reports regarding a drug that is 100% effective at preventing HIV have captured significant public interest. But what is the actual truth behind these headlines? Is it really possible for a medication to offer 100% protection against HIV? The short answer is, yes, it is. A drug known as Lenacapavir, marketed under the brand name Lenca and developed by the American pharmaceutical giant Gilead, has been proven to be 100% effective in preventing HIV infections among women. Here are the specific details of that discovery. In a landmark study published on June 24 in the New England Journal of Medicine, 5,338 women and adolescent girls in Uganda and South Africa were participants in a clinical trial. They were divided into three groups using a 2:2:1 ratio. The first group received injections of Lenacapavir once every 26 weeks. The other two groups were prescribed daily oral doses of either Descovy or Truvada, which are the current standard of care medications for daily HIV prevention. The results were nothing short of extraordinary. Among the group of women and girls who received the Lenacapavir injections, not a single person contracted HIV throughout the entire duration of the study. In stark contrast, approximately 2% of the participants in the groups taking the daily oral pills became infected with the virus. This data confirms that the reports of a 100% effective HIV prevention drug are indeed accurate. However, there are several critical points that we must keep in mind to understand the full context. First, this 100% efficacy rate was specifically observed in women. The biological pathways and risks of HIV transmission can differ significantly between men and women. That being said, medical experts are highly optimistic and confident that Lenacapavir will also demonstrate exceptional preventative results in men once those specific studies are concluded. Second, while the 100% figure is a factual result of this study, it is important to remember the sample size. This result was observed within a specific group of 2,134 women and girls in Uganda and South Africa. When the drug is eventually rolled out for mass use across the globe and applied to a much more diverse population, the efficacy rate might fluctuate slightly and may not remain at a perfect 100% in every real-world scenario. Third, we must look at what this means for the future of medicine. Despite its incredible performance, Lenacapavir is a medication, not an HIV vaccine. There is a fundamental scientific difference between a drug and a vaccine. A vaccine is designed to stimulate the body's own immune system to create long-term protection against a specific pathogen, such as a bacterium or a virus. The general mechanism of a vaccine involves introducing a harmless component of the pathogen, such as a viral protein, a killed virus, or an inactivated version, to train the immune system to recognize and neutralize the actual threat if it is encountered in the future. In contrast, a drug is used to treat, manage, or prevent the symptoms and progression of a disease by directly interacting with the pathogen or the body's biological processes. For example, antibiotics kill bacteria directly, painkillers block pain signals, and insulin helps manage chronic conditions like diabetes. If Lenacapavir were a vaccine, you might only need a single dose or a short series of shots to be protected for life. However, even as a medication, the prospect of receiving a single injection every 6 months and never having to worry about contracting HIV is an incredibly exciting development in public health. Another distinct difference is the timing of administration. Generally, vaccines must be administered well before any exposure to a pathogen occurs. Medications, however, can often be used both before and after exposure. Lenacapavir is no exception to this. It can be administered to individuals who suspect they have been exposed to HIV, such as through the shared use of needles, an accidental needle stick, or unprotected sexual contact with an HIV-positive partner. Furthermore, this drug could be a vital tool for those working in high-risk environments where the threat of HIV exposure is a daily reality. This includes healthcare professionals, as well as workers in the skincare, hair, and nail industries who may come into contact with a client's bodily fluids. In these post-exposure scenarios, the human immunodeficiency virus, HIV, may already be present in the body, entering an incubation period that typically lasts between 1 and 6 weeks. This is where Lenacapavir's unique mechanism of action comes into play. It is classified as a capsid inhibitor. The capsid is the protective protein shell that surrounds and shields the HIV genetic material. A capsid inhibitor works by interfering with this shell in several different ways, all aimed at the primary goal of preventing the virus from replicating. Specifically, lenacapavir disrupts the HIV P24 capsid in three distinct stages: the assembly process, the disassembly process, and the nuclear import process. During the assembly phase, lenacapavir causes the capsid proteins to link together incorrectly. This leads to the formation of malformed, junk, capsids that are unable to properly encase the viral genome as they were designed to do. For the virus to replicate, the HIV genome must be released into the host cell, which requires the capsid shell to break apart or uncoat. During this disassembly phase, lenacapavir actually reinforces the capsid, making it too strong to break open. This effectively traps the viral genetic material inside the shell, preventing its release into the host cell. Finally, for HIV to successfully reproduce and spread, it must smuggle its genome into the cell's nucleus and integrate it into a chromosome. If we think of the viral genome as contraband, the capsid is the smuggler. Lenacapavir prevents the capsid from interacting with the cell's internal transport machinery. When the cell's machinery ignores the capsid, the virus is unable to multiply. However, as the saying goes, nothing this revolutionary comes cheap. Currently, lenacapavir is priced at over $42,000 per year, which is more than 1 billion Vietnamese dong, for the 2 necessary injections in the first year. Health experts have calculated that if the drug were mass-produced for a global market of at least 10 million users annually, the cost could potentially drop to as low as $40 per year. While that is a hopeful vision for the future, we don't yet know when such a price drop might occur. Until lenacapavir becomes more affordable and widely available, it is important to remember that we already have highly effective tools at our disposal. If you look closely at the data from the June 24 report, the daily oral HIV prevention medications, Descovy and Truvada, still boast a protection rate of up to 98%. According to current market data, Descovy costs between $2,100 and $2,300 for a 30-day supply, while Truvada is slightly more accessible at around $2,000 per month. While these are still significant expenses, they represent the current reality of HIV prevention. Speaking of HIV, there has been a recent surge of misinformation online regarding the virus. A fake news story recently went viral about a young woman who was falsely accused of intentionally spreading HIV to dozens of people. Amidst the panic, some social media posts began circulating a claim that the probability of contracting HIV through a single instance of unprotected sexual contact is only 0.08%. The logic presented in these posts suggested that a person would need to have sex hundreds of times before they were at risk of infection. While the 0.08% statistic itself may be derived from certain studies, interpreting and spreading it in this manner is extremely dangerous. It is true that the statistical risk of transmission per individual act of unprotected sex is often cited as being less than 1%, or even as low as 1 in 500. However, this absolutely does not mean that you are safe for the first 99 or 499 times. Statistics don't work that way in the real world. The correct way to understand this probability is as follows: if an HIV-positive individual has unprotected sex with 100 different people, the statistical probability suggests that one of those 100 people will contract the virus. This means that while 99 people might walk away lucky, one person will be the unfortunate individual who is infected on their very first encounter. The logic that you would need 1,600 encounters to infect 16 people is a complete fallacy. You could be one of the lucky ones, or you could be the one who faces a life-changing diagnosis after just one mistake. It is my hope that anyone who encountered those misleading social media posts understands the gravity of the risk. Relying on flawed logic regarding transmission rates is a dangerous game. I hope this video has provided you with valuable and accurate information regarding the latest medical breakthroughs and the realities of HIV prevention.